Okay, we're back again. The structure and regulation of biochemical pathways. Doesn't that sound good? So, this is really about enzyme action, and you've looked at enzyme action again in year 11. We'll do some more with it here in year 12 and hopefully take it a bit further. You should be aware of the term metabolism and hopefully the ideas of anabolic and catabolic metabolism. So, metabolism, everything that happens within chemically, in chemical reactions within an organism, and they can be generally anabolic. They can build things, or they can be catabolic. Now, if you need to remember that, I always think of anabolic steroids building muscle, and this helps me remember which one's which. Photosynthesis is the obvious example of anabolism, and we'll talk about that in great details in a podcast of its own. And catabolism, we'll look at cell respiration. It gets its own podcast as well. And we'll get to those very soon. So, all of these chemical reactions are controlled by enzymes and other proteins, and they work in groups. So we can have a chain of 6 or 7 enzymes interacting with a substrate to create the final product. It's quite a complex process, and we'll try and break it down and make it reasonably straightforward for our purposes. So this is really about enzymes. They're proteins, so enzymes are biological catalysts. I suppose the chemists use enzymes too, don't they? But anyway, we'll talk about our biological ones for now. And essentially, it speeds up a chemical reaction. That's its purpose. Or in many cases, it allows it to happen at all. We know quite a bit about them. They're proteins. They are substrate specific, so they are used on only one thing. Maltase breaks down maltose. That's it. They can be reused. So once that maltase has broken down a molecule of maltose, it's free to break down another molecule. And it'll do that as long as there's substrate around. And therefore we only need them in a small amount, clearly. And that's, again, about energy conservation. Why make more enzyme than you actually need? It's expensive for an organism to do that. They aren't considered reactants or products because they aren't used up in the reaction. They don't take part in forming the new thing we've made. We also know that they reduce the activation energy of a reaction, so it makes it easier for it to occur. They're capable of catalyzing a reaction in either direction. So building a new product or breaking it up. They don't actually change the direction of that reaction though. They go with the flow. And they don't actually change the final amount of product. If you've got 100 grams of substrate, there'll be 100 grams of product. That's all there is to it. Or might be less. You might have wastes, but you won't get more. That's for sure. And they are affected by certain conditions. They have limiting factors. So we'll talk about those in a minute too. There are 2 essential models, and the induced fit model is replacing the lock and key model. And we're seeing much more evidence for that induced fit model being more correct. That's the word I wanted, but I can't think of what it was now. So lock and key model is fairly obvious, and the diagram here shows it quite nicely. We've got this enzyme and a substrate. And the substrate has a particular shape, and on the enzyme there's an active site formed in that tertiary bonding of the protein that fits perfectly to the substrate and allows the two to bind together. Now, induced fit isn't, well, I'm in the way there, aren't I? Come on, if I can move myself. I can. Induced fit isn't that much different. It essentially says, and here's our enzyme here, and there's our 2 substrates. It can wrap itself around those and form the shape of the substrate. So it allows, essentially what we're looking at is, if I was talking about maltose again, maltose is a carbohydrate, a disaccharide, and so maltase may in fact be able to break down other disaccharides that are similar to maltose. It can't break down a protein, it's too dissimilar, but it might break down a small group of things, and this is certainly what we're seeing much more of in more refined research these days. There are 2 major limiting factors on enzymes. The first one is temperature, and hopefully you got this last year. So at low temperatures, and that's actually, that graph's probably bit misdrawn. It probably should be a little bit higher. At very low temperatures, our enzyme will be dormant or acting very, very slowly. And then as more heat energy is available, we see the enzyme start to operate faster until it reaches an optimum temperature. If it goes beyond that optimum temperature, then we actually change the active site, and that's called denaturing the active site, and it won't work anymore, and so we see it fall off in product quite quickly here. So temperature is critical, and you can imagine most enzymes in living, especially in mammals, work best at around the 36 degrees mark, because our core temperature is roughly, 36 to 38 degrees Celsius. As it starts to go beyond 40 degrees Celsius, then our enzymes, our proteins start denaturing, which of course, as you can imagine, is not a good thing. The other one that we tend to look at is the idea of pH. So the pH of the environment that the enzyme is found in can have a major impact. And this time, there is no dormancy. You either are working and your active site's good, or to lower pH, so highly acid, to higher pH, highly alkaline, and in this case, this particular enzyme doesn't work. It's been denatured. And so if we think about our digestive system, which is a classic exam question, in my mouth, I'm slightly alkaline. So the maltase that comes out of my saliva is going to work well in my mouth. As I swallow that food I've been chewing on, and it reaches my stomach is over here. It's 1 pH. That maltase will denature straight away and won't work there anymore. New enzymes will be required. As we leave the stomach and enter the intestine, the intestine goes from reasonably neutral, the mouth is reasonably alkaline to quite alkaline. And again, those enzymes at work will change because the environment's changing. We talk about enzymes having a thing called affinity. So whether or not they can actually lock to their substrate. So we can use chemical inhibition to reduce the affinity. So certain chemicals can be used to slow down the effect of an enzyme. So we might want to control the release of the substrate in some way. Clearly, the amount of reactant available will obviously slow down the speed of it or increase the speed of an enzyme. And we also have things called coenzymes and cofactors, and things like vitamins are coenzymes, and we'll talk more about those too. So affinity is a term used to describe the ease with which an enzyme binds with a substrate. The greater the affinity, the better the binding, the faster the reaction, essentially. So coenzymes are small molecules. Cannot be by themselves used to catalyze a reaction, but they help enzymes to do so. And things like vitamin C, vitamin D, vitamin B, or vitamin Bs, these are all coenzymes, and they work with our enzymes to control the processes. So as a vegetarian, when I eat my sources of iron, I also need to make sure there's vitamin C in that meal, because vitamin C will allow me to uptake iron from my diet. Well, not just being a vegetarian. If you're eating meat and you want the iron from your meat, you should have a source of vitamin C with that. Otherwise, you're not going to be taking up much of that iron to actually pass through. It's very hard to digest iron and get into our blood systems. So some of the classics within our metabolism are things like ATP, NADH, and NADPH. So ATP, obviously you probably know, we'll talk about lots of, is adenosine triphosphate, so it's 3 phosphates present, and it's used in cells to transport energy. So as it releases a phosphate, it becomes ADP, adenosine diphosphate. And of course, it can release another phosphate and become AMP, which we've already met earlier, and we'll talk about much more when we start talking cell signaling. But as it releases a phosphate, it releases energy for the cell. NADH, nicotinamide adenine dinucleotide, you love these names. You can see why we go for NADH. It transports charged hydrogen ions. Really important in photosynthesis and cell respiration, these guys. And we'll talk about those in forthcoming podcasts. And acts as an oxidizing agent within chemical reactions. NADPH, nicotinamide adenine dinucleotide phosphate. So it's NADP, NAD with a P basically. Is also able to carry charged atoms, hydrogen atoms, but it works to reduce. Works as a reducing agent, in chemical reaction. So these three are really important coenzymes in the processes of our cells, and we'll look at those in more detail. Oh, and I finished again. That was another quick one.